Hi, welcome to Detours, Understanding Acquired Brain Injury. We'll be starting a new series. What we want to do is start talking about neurogenetics. Neurogenetics, as if you couldn't tell by the name, looks at how genetics impacts your brain. Specifically, we're interested in how neurogenetics plays a role in both acquired brain injury and how it plays a role in your recovery. But in order to do this, we have to talk about some of the basics. So I won't ask anyone how long it's been since they've taken basic biology, but for some, I suspect it may have been a year or two. And so what we need to do is go over some of the basics of cell biology and genetics in general. So this series will cover essentially an introduction to both cell biology, quick, I promise, the cell biology portion. But we also need to talk about some of the new discoveries when it comes to genetics and mechanisms of chromosomes, how proteins are made, things like that. Because the new discoveries are, they've been made since many of us have been in school. So that's what this is going to be about. Essentially, it's an introduction to contemporary genetics. We want to give you that foundation before we dive into neurogenetics. If you have a solid grounding, of course, in contemporary genetics, you could skip this series, of course. Now, what is contemporary? What do I mean by that? Contemporary is within like the last 5 years. Now, if you've studied within the last 5 years, obviously this series isn't for you, but if it's been longer, I encourage you to follow along. So perhaps you'd like to watch. I do have a lot of this scripted already, so I'll be working with those materials. So let's begin with some things that I'm pretty confident here that you already know, and we'll move from things that you know to things you may not be as familiar with, obviously. Since this is Unit 1, we break it into 3 chapters, and the other units that we move along with will be of varying length, and we'll let you know. And we intend to release these Monday, Wednesday, and Friday. So, and based on the title, you'll be able to tell what we're talking about. So you could skip on ahead or go back and review. Then after we complete genetics, then we'll start looking at neurogenetics more specifically. But that will be a continuing series, and we'll talk about things both developmentally as well as acquired. So, as you know, living things are made of cells. Viruses are an exception, but we aren't talking about them right now. We have spoken in the past about viruses, but it's for now just remember living things are made of cells. The earliest life emerged on Earth about 3.9 billion years ago, give or take a few million. That's fairly soon when it comes to geological time. Early life was mostly in the form of single-celled organisms. The first organisms were aquatic. These organisms lived off a chemical soup from deep volcanic vents. They used these chemicals and the thermal energy to synthesize the proteins that they needed to function. They are referred to as extremophiles. Any organism that lives off of like chemicals and survives in environments that are far beyond what you find on the surface of the planet. These organisms are still around today. Some metabolize sulfuric acid. Some can live in space. There are really some extraordinary living things out there. Some even use ionizing radiation to survive off of. So now, these early organisms which relied on chemical energy far less efficient than photosynthetic organisms which rely on energy from the sun. So, but they still were able to do the things which we consider to live, as far as to synthesize energy from an external source to reproduce. And those are currently the agreed upon definitions for what makes something alive. Early life, as it was progressing over millions of years, really didn't have a single place to store the DNA, the blueprint for life, as it's often called, although you'll learn it's much more than that. The genetic material, the RNA initially, we think, there are other suggestions, was just floating around in its cellular fluid. Cells that function like that don't have an organized place to put the DNA, they still exist today. We call those things prokaryotes. Cells which have developed a place to store their DNA are called eukaryotes. You mean good. Most life on Earth, most single-celled organisms are eukaryotic. Over time, the first single-cell plants, algae, other organisms developed chlorophyll and developed the ability, chlorophyll being stored in these structures which are called chloroplasts. And they were able to pull sunlight and convert it into sugars. And the sugars metabolized into an energy source called ATP, adenosine triphosphate. So this process over time added oxygen to the atmosphere, because early Earth did not have oxygen in the atmosphere. We know from deep core samples and analyzing them, that it didn't. Also, there were other sources of oxygen. We refer to it as deep or dark oxygen, which happen through electrochemical processes, breaking down of certain rocks or reactions, as well as releasing from volcanic sources and other things in the early days, oxygenated the atmosphere. And oxygen is toxic. It has to be broken down in order to prevent it from killing cells. It's why you know products like OxyClean, stuff like that, and free radicals, oxygen-free radicals, have to be safely bound up so they don't destroy DNA, things like that. But we also need oxygen, despite how dangerous it is, in order to live. It drives something called the electron transport chain, which produces from adenosine diphosphate to adenosine triphosphate, which is really what powers cells. So oxygen necessary, but oxygen dangerous too. So it's, as most things that are living, they live on the razor's edge of stability there, and that's all right. So over time, single-celled organisms, they began to live in communities, working together, and with a progression of time, evolution, they developed into tissues, organs, organisms. And, it's this pressure over time, and it is the need to adapt, which led to, all the different critters, dinosaurs, and eventually over time, you got apes, chimps, and us. Okay, so quick 2-cent crash course in evolutionary biology without really talking about the millions and billions of years it took for many subtle changes. And that's what drives the process of evolution. Little tiny changes over long periods of time. And that's what we see when it comes to DNA and how it develops and how changes happen. Very small kinds of changes. Every cell has DNA in it. Chromosomes are genes are on the chromosomes, is what I meant to say. And that's something we definitely will get into, that genes are found on chromosomes. Chromosomes are kind of like a book, and like a thick cookbook, that genes are the recipes that are written into a cookbook. DNA, chromosomes, DNA is the structure for this cookbook into which the recipes are written. So really, like, red blood cells are an example of a cell that does not have in it a nucleus. It has no DNA in it. The progenitor cells from which it comes completes the development of the proteins, replication, and when it's done, it ejects the nucleus, and it lives for 120 days, and that's it. It's the only cell that works like that. But because of its job and because of how short-lived a mature red blood cell is, it doesn't need the nucleus. So there are other cells and other creatures that work like that, but for humans, Now there are other highly specialized cells, neurons, specifically in the central nervous system, and muscle cells, and for production of these cells. While they do have nuclei, they do have DNA, they have to for the production of proteins. They don't divide anymore. They lose certain structures, which we will get into, because they're so highly specialized. It's kind of like, whenever you talk about somebody who's a super specialist in something, that they're really not good at much else in life. Well, this is kind of the same idea. These are ultra specialists. And so, no. So now genes exist primarily to code for proteins day to day. That's their primary function. It's not for division. That's not their primary job. It is to code for all the proteins that are necessary in order to function. Again, think back to when I just mentioned about the cookbook. The old family cookbook, there are certain things you want to make more than every single thing that's in that book. Maybe grandma's favorite chocolate chip cookie recipe is the one you really like to use, or lasagna, mom's lasagna, or whatever else. Those recipes are frequently used, whereas, great aunt, Bertie's fruitcake recipe, it doesn't get used that often. And even rarer is that you copy the entire family cookbook to give to your kids or, your nieces or whatever. So keep that in mind. The main job of DNA is to copy for the proteins you need day to day. Okay? Just kind of stick a pin in that. So for now, all of this is less important at this moment to discuss, but I did want you to think about that the main job of the DNA on the chromosomes is to code for everyday working proteins. For division, replication, for having a baby, whatever else, that is not something that happens commonly. It is to code for proteins day to day, to get those recipes out that you use frequently. Okay? That. And for now, what we're going to do is we're going to take a break. And this is the end of chapter 1, just an introduction to what we're going to be talking about. And chapter 2, we will be moving on to the organelles, the machinery inside the cell that is necessary in order to produce proteins. And we'll be getting properly into that in Unit 2. But for now, just a quick overview. So please like, subscribe, don't forget to hit the bell icon. And so we will see you next time. And please share. And please, any questions or anything that you have, please let us know. That's important to help us with further development or to help us to make the necessary corrections. Bye. See you soon.